Hello, my name is Stella CHRISTOU and I'm a PhD student in environmental engineering at the University of Surrey. I received an EBNet bursary to attend EDAR7 conference, which stands for Environmental Dimension of Antimicrobial Resistance, and this year was held in Montreal, Canada. And there I gave an oral presentation, which I'm very happy to share with you. My work is about exploring antimicrobial resistance in conventional and advanced wastewater treatment processes, and it is a cross-study along the oxygen gradient. So the fundamental role of wastewater treatment plants is to receive wastewater discharge, treat it through different stages, and then return it to the water bodies. And by treatment, we mean the removal of organic matter, nutrients, pathogens, pollutants, chemicals, etc. And when we refer to secondary treatment, we mainly mean biological treatment, where microbial communities perform the aforementioned treatment of wastewater, while they're also exposed to other contaminants such as antibiotic-resistant bacteria, antibiotics, antibiotic-resistance genes, and other pollutants that altogether create suitable conditions for antibiotic resistance dissemination and further spread to the environment through treated wastewater, as wastewater treatment plants are not designed to remove antimicrobial resistance. During the past years, population growth, high pollutant load discharge, and rising wastewater generation have created the need for wastewater treatment plants advancement within the context of circular economy. And this means that not only they need to provide clean water, but they should also strive for water recycling, resource recovery, renewable energy generation, and energy reduction. The latter is very important since 1 to 3% of global energy is consumed on wastewater treatment plants, with aeration being the most energy demanding process, accounting for 75% of conventional wastewater treatment plants. And how can this transition look like? It could be a shift to a different biological treatment method, such as aerobic granular sludge, where its compact infrastructure results to lower land footprint and lower investment costs. It consumes less energy while providing better effluent quality. There is lower sludge production and resource recovery potential. But in reality, it could also be an addition of treatment units to an already existing conventional process, such as an aerobic digestion unit, which could also result to biogas production, or partial nitrification Anammox tank that could help with the carbon recirculation. So the aim of this cross-study is to examine antimicrobial resistance profiles in conventional and advanced treatment tanks that are distinguished by the oxygen factor, which makes them aerobic or anaerobic. To achieve this, we analyzed in-tank data from antibiotic resistance genes and mobile genetic elements from 3 different studies: anaerobic digestion treating black water with microaeration, aerobic granular sludge treating wastewater, and the treatment line with anoxic aeration, anaerobic digestion, and partial nitrification Anammox treatment tanks. From all studies, data originated from metagenomics and or qPCR. For the first study, some background is that conventional anaerobic digestion and generally is performed in high temperatures, which results to high cost and energy consumption. And the low-cost strategy for this is microaeration to enhance in situ hydrolysis in a single tank. In this study was a lab simulation treating black water, which holds the capacity for biomethane production, and the data originated from qPCR. Different oxygen input from 0 to 150 milligram per gram COD feed was examined. The results of this study have shown that in low oxygen, methanogenesis efficiency is higher, and that all antibiotic resistance genes and mobile genetic elements are more abundant, with a minimum average of 2.3 times higher than in high oxygen conditions. A ANOVA test revealed significant correlation of oxygen dosage to antibiotic resistance genes and mobile genetic elements quantity. And the mobile genetic elements abundance in the feed black water was significantly low, indicating that microaeration in low doses could be the driver of their increase. In literature, it was shown that horizontal gene transfer frequency and plasmid retention are enhanced in low oxygen conditions. The second study concerned an aerobic granular sludge single tank process, and samples were taken from 4 different stages, but for the scope of this study, samples from the beginning and end of aeration were used. Aeration lasted approximately 9 hours, and the DNA samples that we used for metagenomics and qPCR concern intracellular and extracellular DNA, where intracellular DNA originated from different sized granular sludge and flocculent sludge. The results of the metagenomic analysis indicated that in aerobic granular sludge, pre and post aeration main antibiotic resistances did not significantly change, and the main resistance groups were MLS, aminoglycosides, sulfonamides, beta-lactam, and tetracycline. The extracellular DNA resistance profiles at the beginning and end of aeration resembled the intracellular DNA resistance of the granules. And from the taxonomic analysis, it was shown that extracellular antibiotic resistance genes mainly originated from the granules rather than from the flux. From the qPCR, it was shown that there were no significant differences between granules at the beginning of aeration, but post-aeration, small granules accumulated more resistance than large granules and flux, and this is possibly due to higher immigration capacity and shorter solid retention time of small granules compared to the large ones. And finally, the third study concerned an advanced treatment line with anoxic aeration, anaerobic digestion, and partial nitrification Anammox tanks treating wastewater. In this case, data originated from metagenomics, but since this study is still not published, the data were excluded from this presentation, but some results are included in the conclusions part. To conclude, different anaerobic digestion processes demonstrate opposing antibiotic resistance genes abundance trends in low or no oxygen conditions, and this is possibly due to thermal hydrolysis pretreatment that is included in the latest study. Oxygen is not the only parameter that can potentially affect antimicrobial resistance. Advanced partial nitrification Anammox treatment tank demonstrated significantly higher antibiotic resistant genes accumulation. Aeration does not significantly change antibiotic resistance genes accumulation in the aerobic granular sludge treatment, and the increase in small granules post-aeration may indicate a route for AMR dissemination. No or low oxygen conditions potentially influence mobile genetic elements accumulation, so processes employing passive aeration should be explored. And finally, a firm conclusion on the effect of oxygen supply on antimicrobial resistance load cannot be derived from these studies, but this study triggers more hypothesis tests on pollution control. I would like to thank the co-authors of this study, David Calderon Franco, McAnthony Norum, Jennifer Ritchie, Yang Liu, Mark van Lustrecht, David Weisbrod, and my supervisor Bing Guo. And finally, I would like to thank EBINET for the bursary and the opportunity to travel to EDAR7 conference. This was very beneficial for my PhD project, but also for my professional development.